Today, we explore the mechanisms of cellular memory and gene regulation. Our guest, Dr. Eva Hermanns-Eder from the Helmholtz Zentrum in Munich, investigates how cells preserve their identity through mitotic divisions, an essential process for development and disease. By studying chromatin dynamics and epigenetic modifications, her research sheds light on how genes are switched on and off over time, with major implications for cancer biology, regenerative medicine, and beyond.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Today I'm not at home in my home office and doing this recording over Zoom, but today I'm actually in a lab or in your office, Eva, and I'm happy to talk to you face to face.
Hello, Stefan. I'm also very happy to. I'm curious what is going to happen next.
Yeah, it's going to be a conversation about your work. And a question I like to ask every guest to ease into the topic would be, how did you become interested in biology in the first place and then pursuing a career in science?
How I became interested? I think it was still in high school when I was a pupil. I had to search for a topic for my final thesis that we had to write at that point. And what I found the most curious thing was how this egg, when fertilized, how it divides and how it becomes all these different cells, how you can become from one, many. And somehow this started to... From this, I learned that I have to go into developmental biology, into genetics to understand that further. And initially, I wanted to become teacher, actually a high school teacher. But I was so curious and I was so drawn by all these developmental biology lectures that I always just continued until I did my master's thesis with Jürgen Knoblich in the IMBA in Vienna at that time, and continued to look at these early embryonic cell divisions from my doctoral thesis in Constance, and then again going to understand what this memory of a cell is and how it's established. I went to John Gurdon then in Cambridge. So this was always a driving question for me, actually, since I'm 17 years old.
Have you been aware from the beginning what this would mean, having an academic career?
I remember that when I started, everybody told me after your master's, there's no chance you're gonna get a job. It's absolutely horrendous and it will be impossible. So at that time, I was like, yeah, I'm doing something where it's very difficult to find a job afterwards. And actually, I didn't find that to be true. I think it depends on how determined or focused you are on one specific thing. If you have exactly one thing in mind and you have no flexibility, then I think it's probably hard. But if you're a bit flexible about what you want to do, which question you want to ask, and if you want to go in industry or anything, I think it's actually quite promising. So of academia, I expected, nothing, because I'm actually the first in my family that is doing anything like that. So I had zero experience. I did not know anything.
So you are still in the early stages of your research career. I guess one can safely say that. And you are now at the Helmholtz Zentrum Munich. Maybe can you talk about how you got here and what's maybe your plan moving forward?
So how I got here so I had this paper in 2017 about H3K4 trimethylation, and that it's probably an epigenetic mark for active transcriptional states. So this is also something that we try to still investigate and to prove or to, underline that this is really an active epigenetic mark, because there's so much debate about it, and, we want to show the propagation and the function and everything. And so, but I gave a talk about this at the conference. I think it was the Chromatin conference from Epchem. Sorry.
Yes.
And I gave a talk and somehow there I connected with a lot of people that then afterwards offered me to apply to the institute. And one of the people that offered me this position, or this possibility, not a position obviously, was Maria Elena. And then she invited me for an interview, and then I came here, and then I was just blown away by the community here. The chromatin field is amazing here. The conversations I had after my talk, the one-to-ones, the questions they asked and the possibilities, I was extremely excited about it, and I thought, this is the place where I want to go. This is where I want to be. And somehow I then did not consider other places anymore and just came here when I got the offer.
Yeah, that's obviously what you do then, right? If you know that this is the right place, then
you feel it also when you are there.
So let's come to your science that centers around epigenetic memory, stem cell reprogramming, and barriers for cell fate changes. I want to start in the year 2017. Then you were with Jérôme Julien, hope I pronounced this correctly, and working on nuclear transfer embryos and how they retain their transcriptional memory. In a paper that was published in this year that I mentioned, you looked at the role of what you just mentioned, right? H3K4 methylation in transcriptional reprogramming. Can you talk about the study? I guess this was in your postdoc, if I'm correct.
So I think one point I need to correct you quickly. So it was the lab of John Gurdon, where both Jérôme and we were postdoc students there. And I don't know why it ended up that Jérôme was the last author, because I think he supported me a lot with writing and everything, and it was obviously fair that he would be the last co-corresponding author as well. But I think it also shows you one remarkable thing about John Gurdon is that he was the one that would propose it, and I think this is something that is so different for him than of any other or many other people. I mean, the people that I had in my career, which is Jürgen and Thomas Meyer, both of them always supported me. But with him, he has people, he gives a space in your lab, and then you can do your experiments. But then he makes it clear that everything you produce is yours. So I could take it with me. I could work on all the projects that I developed there. And also when it came then to the publication, he's very humble and he wants to promote you and your career. So he also steps back there then and said, look, I actually did not contribute that much. I think it would be fair if Jérôme would be here the last author. And for us, this was also a perfect solution. So in the end, it was John and Jérôme being the last author. But I think it's a nice example how this lab worked and how his way of science was. And I think this is also how I was taught, by being curiosity-driven, to ask a question. And he would always ask, what was the best case scenario of your outcome? And when I started in his lab, I saw that in nuclear reprogramming, everybody focused on these repressive genes that were not properly activated during reprogramming. And at that time, when I started, people said that active genes, when I asked, so how are now active genes inactivated? I came from the cell cycle. I had no idea about chromatin and transcript. So I asked, so how are then active genes inactivated? And nobody could give me an adequate answer. Everybody says, yeah, transcription factors go away and eventually, somehow just stops. There was then later work on enhancer decommissioning and so on, but at that time nobody really was giving me a proper answer. And then I wanted to address in the nuclear transfer whether that is at all a problem during reprogramming. So we did this first experiment where we did gene expression analysis, and then we saw that in the frog actually the genes that were not inactivated and were specific of the starting cell type of reprogramming, that these genes were as many and the phenotype was as strong as the genes that were not properly activated or upregulated during reprogramming. So, and then I realized that it could be a problem. And when I looked at these genes, they were really highly enriched into lineage genes. And we used endoderm to ectoderm reprogramming. And it was key endoderm transcription factors that were always active in the embryos. And so from there, I thought, there might be a problem. And it didn't correlate with the gene expression, and also the transcription factors that regulate those genes. They are not present in the egg. And you dilute 1 in 10,000 when you transfer. And then also this cell divides, this genome divides another 12 times before you activate gene expression. So we didn't really think it's transcription factors that contribute this maintenance. So we went towards chromatin marks and epigenetic mechanisms. And one key active mark was H3K4 trimethylation. And we had some indication from previous experiments in the lab that it could have a role, but we didn't address it genome-wide and on the rescue experiments, and so then we went towards that one, and I was lucky. But I tried many other things first before I found something that was good. I tried the 3D genome architecture and everything. So I did many experiments, but finally with H3K4me3 we found something nice. And really when we removed that, we saw that these active genes were suddenly downregulated. So there is a regulation of this active mark, and the reprogramming efficiency became really, really high, almost like if you would take undifferentiated blastocyst nuclei to make the nuclear transfer embryo. So it really suggests that H3K4me3 is a barrier to cell fate changes.
So I guess we will come to that later, but maybe I can ask here. Because did you also look, you see that it goes away, but what other factors then come in to change the transcriptional landscape? Because if H3K4 methylation goes away, then you might think to deactivate and to silence the gene, there must be something else coming in and putting marks there. So did you look at that, or is it something that you're looking at?
No, I think that is a really important question. And just now was Antoine Peter's talk, who also looks at how genes are then, how important it is to keep away silencing activities. So we have several theories, because later on we also found that now, this is the paper that we have currently under revisions, where we also see that H3K4 methylation seems to be important also for zygotic genome activation. And we like to think, and this is a lot of discussions, whether it's maintained, propagated, transmitted, continuously propagated. But I can tell you, we see that H3K4me3 is present on promoters in the spermatid, that is transcriptionally active. It's still at the promoter in the sperm. It's still at the promoters in pre-CGA embryos. There is no transcription yet, and still 12 cell divisions. After 8 divisions, it's still there. If it was continuously maintained, we don't know, because it could be a half an hour window where it was gone. But we see it's still there, and at CGA it's also there. And when we remove it, we get the defect in zygotic genome activation, and preferentially of genes that had kept it. So we like to think that H3K4me3 maintenance is important for CGA and for the proper expression of CGA transcription factors. But is it because if you look at Christian Helin's work, is it promoting efficient transcription at CGA? Because you need that in the gene body, and we also see it's preferentially broad domains that go in the gene body. Or is it because it prevents DNA methylation? We don't know. So in the end, it could be that one pathway of this inactivation, that you allow inactivation actually, that you don't actively maintain something, but you allow inactivation when you remove H3K4 trimethylation. But I do not know what happens to other things like H3K36 dimethylation, and whether now DNA methylation increases. This we don't know.
So when you go from 1 to the 4 stage, you have 4 cell divisions, so you would dilute, if I'm doing the math correct, 8 cell divisions. So you would dilute it by a factor that I can't calculate now. So you must have an enzyme that puts it there, right?
Yeah. So there are 256 nuclei after. So you start with one nucleus and then you enter 256 nuclei. So one thing, I think one big drawback that we have in terms of normal development, whether this is important in frog, is one that we have no idea how the oocyte looks like. And would be very good if we are trying right now actually to get a cut and run to work to get the information of the oocyte, because in frog that's completely a mystery right now. So we have only the information of the male. So I can tell you from the male offspring, we can see that these domains, they are very large, they can be several kbs, but still if you calculate a 1-to-1 dilution at every cell division, after this 8 cell division, it should be theoretically gone if there is no active maintenance.
Yeah.
But whether it's promoter specific, if there is it could be that the maternal genome is completely methylated and then reduced to this direction. This we don't know.
Okay. So after that, I guess you transferred to your own lab, and in January this year, so we are in 2025, 3 papers were uploaded to bioRxiv, and the first one, which is now published in Stem Cell Reports since last week, which was March 13th, you showed that the differentiation success of reprogrammed cells varies across cell types and depends on the expression of genes specific to the previous cellular identity. Can you talk about maybe the transition from your postdoc lab to your own lab, and then how this work started and what you found there?
So I think one question was then really, we thought about these transcription factors. Once they are there, how long does this effect last of these transcription factors? How long can we observe endoderm transcription factors in ectoderm cells? Is every cell type equally affected by these problems, because an embryo, what we measure reprogramming efficiency is by embryonic death, but the embryo could die because of many different things. So we wanted to go a bit more specific and not just score embryonic death, but we wanted to look how well can we make a differentiated tissue out of reprogrammed cells. And in nuclear transfer embryo, nobody has looked at that, surprisingly. People look usually at the 2-cell or 4-cell or 8-cell stage and then death. but nobody looks actually whether then the differentiation works properly. So when we went in this project, we used single cell sequencing because it was possible at that time. And so we started that experiment, and we expected that we get a clear separation from nuclear transfer cells versus IVF cells. And what we in fact found is that the nuclear transfers or the reprogrammed cells were overlapping or co-clustering with the cells from the fertilized embryos, or the proper counterpart. And that was surprising, because we knew that out of this embryo that we sequenced, 70 or 80% would die. So we thought that at least 70 to 80% of these cells should have massive defects. But we found that it was actually only some groups of clusters or cell types that showed defects. And I think this was also a possibility because we had a proper wild type counterpart. When you normally do transdifferentiation, you never have the real backdrop of, or rarely, or in the studies that I know, you don't have a proper backdrop of what it's supposed to be. So usually you depend on certain markers. And we didn't, because we had the proper dynamics and the transcriptome of the wild type background, and we compared it to those, and then we found that they showed in certain cell type defects in the dynamics and the differentiation dynamics, and also an underrepresentation of certain cell types. And then we found that, for example, the goblet cells of this epithelial cell layer was perfectly fine, but the basal stem cell and their progeny had massive differentiation defects. And also we observed apoptosis, and we could rescue that very defect by reducing the expression of one on-memory transcription factor. So we reduced it specifically by injecting a morpholino into a certain cell, and that gives rise to the epithelium. And then we could rescue this defect there in the epithelium. Mm-hmm. And that was really nice for us, because this is something that was, for me, completely unexpected, that there is only a very cell type specific phenotype.
Mm-hmm. Did you also look at mechanistical things?
Like
3D organization of the genome or histone marks?
Yeah, so at the 3D, so there were some, so I started off in one of my postdoc projects was also actually to look at the contribution of 3D genome organization to reprogramming. And it was that there was Rick Kelly's thought who saw that mitotic cells were more prone to reprogramming in the lab. I thought, okay, mitotic cells also lost 3D genome architecture. This was just a paper that came out at that time where they showed that 3D genome architecture was removed or lost in mitosis. And from there, I started looking at this. But when we then took, in a collaboration with the Merkenschlager lab, we looked at the REC8 deficient, so cohesin deficient nuclei of lymphocytes at that time, and transplanted them to the frog oocyte, which is another reprogramming system. And then we saw that these cells were just normally reprogrammed, and we saw no defect, which in the end, luckily, we could publish it thanks to Matthias Merkenschlager, but we didn't see any phenotype. But then other people, what we did not look at, we looked at whether it affects the activation of genes, but I never looked at actually whether it maintains gene expression preferentially. So this active genes, where they would stabilize gene expression, this I never looked at then, and was also very difficult at that time to look at this. But there's still a possibility for that. There's a paper about the 3D genome architecture influence reprogramming activities, but also they interfere with it before reprogramming, and then you see already changes in gene expression, and then you reprogram better. But a functional mechanistic analysis, if you would interfere with the 3D structure, transplant the nucleus, look at the embryo, and see whether then really, the maintenance of active states is lost, this is missing. And
I guess it's also very hard to find like a causal relationship between genome architecture and like transcription or anything downstream. It's just like if you manipulate anything downstream, then you can look at 3D structure, but if it's really a causal relationship, I guess it's really hard to find, right?
I think one thing would be nice in the frog system would be very useful, because we have some key candidate genes like SOC17 or GATA6 that are always showing a lot of on-memory. They seem to be enhancer regulated, and we did 3D chromatin 3D structure with Vakarissa's lab and with Sarah, and we see that some of these genes are in structured regions, and we would need to, look at specific genes. I think if there's Boyan, with who you will talk later on, he has a lot of cool tools how you could interfere with certain chromatin 3D structure enhancer promoter interactions. So we could look at a few candidate on-memory gene where we specifically interrupt them, see whether it affects 3D structure and whether that has then a result on the expression. But I think people will not give me that much money because I'm junior, and I have no experience in 3D structure. And I don't think any grant body would give me that money to do those experiments.
So the second paper in bioRxiv was about the H3K4 methylation, and we already talked about that.
Yes, exactly. We talked about this because one thing that was intriguing that we saw this maintenance of active states in nuclear transfer embryo, and that it was H3K4me3 dependent at some point. And we didn't think that this was invented by the oocyte and the embryo to make our life harder to reprogram cells. So we thought it must be there should be some natural mechanism for that. There should be a role for that in normal development, and that's why the frog has it. So we actually went to look for exactly such genes like these on-memory genes, but now in the spermatid. Or in the sperm. Because we thought if this is a feature of such genes, and in this other study that I guess we talk later in digital reprogramming, we also saw that these on-memory genes are defined by H3K4me3, high CG rich, low DNA methylation, H3K27 acetylation on the enhancer, and accessibility. And we thought, okay, this is a kind of a DNA sequence and chromatin setup that could be indicative of, or could be something that is copied and maintained. And also, it's bona fide template for CXXC1, which is CFP1, which binds an unmethylated CG-rich promoters and propagates H3K4 trimethylation. So from this point, we went on in the frog and looked whether H3K4 is at all present, because experiments were lacking to really address whether this mark was present prior to zygotic genome activation. Some people inhibited transcription and then looked at zygotic genome activation, where alpha-amanitin inhibited embryos were used, and used this as a proxy. But nobody looked at actual earlier embryos where there's really no transcription. And after this, whether I also thought, is it diluted or not? And I think maybe the breath helps it to not be diluted. So we looked there with chip, and then we were said, but it's an antibody that just binds anything if it doesn't find a target. It's an artifact. So then we went on doing Western blots, which is still antibody, but Meghana, who was the first one that was able to produce nuclei from mass spec in the frog embryos. So she really established a method to purify nuclei from these pre-CGA embryos and to send them to mass spec for the histone tails. And then we actually found that this pre-CGA nuclei are highly decorated by chromatin marks, including H3K4 trimethylation, and di and mono. So this was the starting point, that it's worth looking at that further, right? And then we did lots of, analysis where we looked at is there something that is always there, like I explained you earlier, and then we started to interfere with potential mechanisms to change that with this KMT2B and CXXC1, because the enzymes had to be transcription independent, because there is no transcription, right? And this was shown to be transcription independent in the oocyte, but never, if it's important, they never addressed whether they also would propagate something, in dividing cells. So we tested it, and we saw that when we deplete them, we see a reduction of H3K4 trimethylation and also a zygotic genome effect. But then we did not know, is it important for zygotic genome activation or is it important for the propagation? Because there is no doubt that H3K4me3 at zygotic genome activation is important. But this could be just, to allow the transcription. And it could be that it's the de novo establishment at CGA that is important. So one very key killer experiment was very difficult to set up. It was that we had a time window interference with H3K4me3. And this is also, again, Meghana's work, because she never gave up on that. She's very resilient. So she managed to express H3K4 demethylase under the auxin decron system, right? So what she could do, she injected this enzyme, let it be expressed. H3K4me3 went down until the 256 cell stage, so after 8 cell divisions, and then she degraded those enzymes or these proteins that interfered with H3K4 trimethylation. So, and then checked that H3K4me3 would go back up normally, which she saw. And then she analyzed this by gene expression analysis and the developmental outcome. So gene expression analysis is the first thing we got. And when we interfered with H3K4me3 in only this small time window of development before CGA, we saw a CGA phenotype. And that was almost as strong as if we would interfere with it throughout continuous, also over CGA. And we also saw that one of the effectors gene were these zygotic genome activation enzymes, transcription factors that were described by George Gage, which is a Pau factor, a OCT4 relative, and SOX2. They are downregulated, and we speculate that this is why there is this overlap, and there's also a lot of housekeeping genes that were affected. So we think there's H3K4me3 might be important for, you don't get a CGA block, but I think it's important for a proper CGA, because it's really strong and it's really dynamic, the CGA, and that this takes place in the correct dynamics. I think H3K4me3 is important.
So coming to the third paper, and which would be the last, you developed an artificial intelligence-based approach to model nuclear reprogramming and used it to identify the chromatin modification H3K27 acetyl as an epigenetic barrier to reprogramming-induced cell fate changes. Could you describe how AI was integrated into this study and what you found?
I think so. First, I did not develop that. I did just tons and tons of dissections to do all the histomarks. I think I dissected several thousand embryos to get all these histomarks, because what we found to predict or to specify on-memory genes, it's very important that you have the exact cell type. We cannot just go to Xencode and get some. You need to really have exactly the correct cell type for the active marks, because only there they will correlate with the genes. So I dissected tons and tons of embryo to get all these active marks chips, and then I was so lucky that there was Chris Penfold, who was just looking for a lab, joined our lab, and I had some funding available, that because John allowed me to apply for third-party funding, so it all worked together. That Chris then joined our lab, and he happened to be a real good computational biologist and highly trained in machine learning approaches, and he was like, oh, let's look at this with convolutional neural networks. And I thought it was about neurons, and I didn't understand. And he explained me everything, how it works, and that it's artificial intelligence, and that the computer can find patterns there in this data. And he basically looked into this, and he taught me also how all of this works. And one funny thing is, I think we would have gotten to all the epigenetic barriers also if we would have done the standard, right? You have the resistant genes, you do blot, and then you see that they are enriched for these histomarks. But what we saw is more that we understood more, first of all, a grammar of histomarks. We found that there are subgroups of genes that are explained by one histomark, subgroups by many, and subgroups by none. This is one thing. And the other thing is, I found it remarkable that a computer could predict reprogramming outcomes. We fed the computer one reprogramming setup, like endoderm to ectoderm, and it, with whatever information it found, it was able to predict mesoderm to endoderm, mesoderm to ectoderm reprogramming without. So of course we did the experiment to confirm that it was able to. But in theory, you could use digital reprogramming, this is how we called our system, to predict any reprogramming outcome without doing the actual experiment. And that would save you a lot of time. So this is something I think that is really remarkable, that you can model reprogramming. Using chromatin and transcriptome data, you can model what's going to happen to the genes during reprogramming. And we could also use it to identify some of the epigenetic barriers using deep explain. So it's another algorithm that can look in the black box of this convolutional neural network.
So was this there have been many people and many systems that describe the chromatin landscape, you have 14 types of chromatin, with different colors or different types. Were those similar to those states, or didn't you check?
I'm not sure I understand what you mean.
There was this Chrome HMM, right, where you have the different chromatin colors, like black chromatin or inactive chromatin, and then there was poised chromatin and active chromatin and even deep silenced chromatin, right?
So I think this. So we started also now to look at, but we stopped now, but we started to look also in other reprogramming systems where they used highly differentiated cells. So I have to say a disclaimer, our cells that we used are not highly differentiated cells. They are already fully differentiated. They are differentiated committed cells, but not terminally differentiated cells. So we don't have the proper somatic setup of chromatin. We do not have there. Yet, they have already reprogramming resistance. John did this nice experiment where you could see that a blastocyst had 60% survival rate, and then where we are is 20% survival rate, and then you go down to 5 or 3% with more differentiated cells. So we looked at a bit better one in order that we had something to look at. Otherwise, everything would be dead, right? So So this is the disclaimer. So that's why I think we might not find one-to-one the real somatic, setup of chromatin. But I don't want to give an answer to this because I would have to speculate too much.
So is this paper also in review?
This paper is also in revisions right now, yes.
And can we expect it to be out sometime soon?
Yeah, this you have to tell our reviewers.
Okay. So imagine you're due to submitting a grant proposal tomorrow. What would you have written into this?
So I think there are two things I find extremely curious is like this first question that you asked me about how are these active genes once we remove H3K4me3, how are they actually silenced? So this how is it transitioning from an active to an inactive state? And also what's the role of the different marks during embryonic development? How are they established? How much does the breadth of a mark contribute? Is it really K27 at the enhancer that matters? Can we manipulate somehow, go more into the nitty-gritty of all of these marks? I mean, if I would look just at my curiosity, not at impact factors of the paper, I think I would really like to discern this, these intricate relationships, because also it seems sometimes in frog a bit different because we see H3K4me3 and DNA methylation on some genes, but are they on the same? There are lots of questions that you could address there. And the other thing that I think is really understudied is so we always say we use reprogrammed cells. We study this because eventually we would like to enhance cell replacement therapies. And I think one big problem still is once you have the reprogrammed cells, you need to integrate them into the tissue or you have to generate the tissue where the function is lost. And I think that is really difficult still because either you have to amplify the cells a lot if you go towards the IPS route, and there you accumulate a lot of tumor mutations, tumor driving mutation, and you accumulate mutations in genes that have fast division and survival and can outcompete other cells. So this is not necessarily the best thing. And also then when you transplant differentiated cells, also that you make from normal ES cells to replace endogenous cell, the integration rate is very low and most of them die or are kicked out by the tissue. And I feel that this is not fully understood. And I think the frog model would be great to address it because it's accessible during tissue differentiation. It lives in the tap water. So you can look at it, and it also has organoids models. So you could, for example, do this experiment by taking reprogrammed cells from nuclear transfer embryo, disaggregate the embryo and reaggregate them, and then see how do the different cells contribute. Are the reprogrammed cells kicked out? Are they killed? Are they just, extruded or something? So why is it that the reprogrammed cells cannot integrate? And those that integrate, which ones are those? And so then to improve that actually, and for the basic research of this question, I think the frog model would be very good to address.
So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think in all this career, at least since now that I have a lab, I was merely an enabler. I think the power was really from the people. I was extremely lucky with the people that I got in the beginning. I have Marco, Anna, and Megana, and they were amazing students. I was also on maternity leave. I was gone half a year. We had to go through COVID. We had to go through a cyber attack. So there was, I think in this time I was here, there was at least almost 2 years where we did not run on full capacity. And then we had frog license issues because the frog Xenopus got under invasive species, and we had to ask a frog license. So for that, it was really crucial that they never gave up and they always pushed and they did a lot. And I did not do much to that actually, so I just managed to be a platform for them. And the other thing is that was very beneficial for my work was the environment or the community of Xenopus, and for example also for the single cell sequencing work. We got so much support by Leon Peshkin and by Ksenia Kuznetsova. I hope I said her name properly. So they are really, she was a genius. She did so much. She helped us so much with cell type assignment to the atlas, to the frog atlas. So, and also in general, the Xenopus community was always supportive and nice. There are too many to name now, but all Xenopus people in the end.
So my last question would be, what is your personal definition of epigenetics?
I like best the one that was, where was this one published first? Leonie Ringross, I think, had this in one of her reviews. It's a state that, so epigenetic state is a state that is inherited independent of the signal that induced it. So I think what I would consider as epigenetics could be is an epigenetic state, is something that is on top of the genome that was induced at one point in development, like, for example, in our case, during spermatogenesis. And then these factors that induce this are gone, and it's a mark on the chromatin, and that is then propagated. So, and there are mechanisms to copy it, okay? But these mechanisms don't have to be only chromatin modifications. I would also argue that there are, like, for example, for our active state, I don't think that H3K4me3 is doing it all, right? So I don't know, strictly speaking, if you call it, then some people might not call it epigenetic, because I think the underlying DNA sequence is crucial without that. So I think if you remove it, the sequence alone would be enough to re-establish it. The absence of DNA methylation is important. There could be transcription factors, there could be DNA modifiers that also bind there. So I think it's a whole protein complex of transcription factor, epigenetic modulators that is copied independent of the signal that induced it, and also independent of, so it's also not only just because it's re-emerged because transcription is continuous. You know, if the transcription factor would be always present to transcribe it and transcribe it, I would also say it's not epigenetic, but there's no more transcription and still this setup is somehow propagated, this active state is propagated. So I would call this is also epigenetic. And I do not I agree with all the people that say DNA methylation is epigenetic. So I have a bit of a looser definition, I guess.
Okay, thank you, Eva, for your time and for being on this show.
Thank you as well.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.